Okay, so Lab 2 was about taxonomy. Taxonomy is the science of naming and classifying organisms, usually according to how closely related they are to each other. And so first what I'm going to do is going to talk a little bit about how to write scientific names correctly and also how to abbreviate them. So a binomial scientific name is made up of two parts. The first part is the genus name. A genus name is followed by the specific epithet. And it's always written in a specific way, and I will show you an example. Okay, so Homo sapiens is the scientific name of human beings. It's got two parts, the genus name, followed by the specific epithet. The genus name is always capitalized. The specific epithet is always in lowercase. So this is our species. Now, it is possible to subdivide a species into what's called a subspecies. Subspecies have only small differences from each other. For example, maybe they're mice that have different colors. Mice in a population have slightly different color fur from each other. They're all members of the same species, but they're just slightly different. Scientists want to give them a particular name so they can be identified. A subspecies name is tacked on to the end of the binomial scientific name. So it turns the binomial name into a trinomial name. So in our manual, we've looked at different subspecies of ringneck snakes. For example, we looked at the Didelphis punctatus regalis, the regal ringneck snake. Didelphis is the genus name. Punctatus is the specific epithet. It's followed by the third subspecies name. So if you're talking about ringneck snakes in general, you're talking about Didelphis punctatus. That's the scientific name. If you want to specify it's for the regal ringneck snake, you would tack on that third name. Now, once again, the genus must always be capitalized, and the specific epithet and the subspecies name have to be always in lowercase. And scientific names must always be underlined, like I've done here, or done in italics. Okay. Now, it is possible to abbreviate scientific names under certain circumstances. If you have just a binomial name like Homo sapiens, you can abbreviate it only if you've repeated the same genus twice in a row. First time you always have to write it out. The second time you write it, you can abbreviate the genus in this manner. Abbreviate the genus with first initial followed by a period. You write out the subspecies, or excuse me, the specific epithet. Okay. And if you go ahead and mention the genus, write out the whole name, then mention the genus again, you can abbreviate it. But if you mention a different genus, and then mention the first genus again, you have to write out the first genus, because you can only abbreviate a genus if you mention it consecutively. You can abbreviate subspecies names in a similar manner. The difference is when you're abbreviating a subspecies name, you can now abbreviate both the genus and the specific epithet if you have the genus and specific epithet repeated. So if you had Didelphis punctatus regalis mentioned, you would have to write it out first. And if you went and mentioned, for example, Didelphis punctatus edgewordi, that is the northern ringneck snake, you could abbreviate DP edgewordi, like so. Okay, so the genus and specific epithet are repeated, so they can be abbreviated, and you just have to always write out the subspecies name no matter what. Of course, it needs to be underlined. Okay, and once again, if you were to mention a different genus following, you need to write out the first one again, you have to write it out again. It can only be mentioned consecutively. You can only abbreviate it if you write it consecutively. So that's the basic rules for abbreviating scientific names. And next, what we want to do is we want to go over just a little bit about taxonomy. I would like for you to remember the taxonomic scheme in order from largest groups to smallest groups. And it goes from domain, which is the biggest group, followed by kingdom, phylum, class, order, family, genus, and species. And that's in your book. And there's a simple phrase you can use to remember that, and that is King Philip came up with a great stake. Stands for domain, kingdom, phylum, class, order, family, genus, and species. So in this lab, you do not have to memorize all the kingdoms and all the phylums and all of that. You're going to be doing that in Principles 2. For our purposes, what I want you to do is be able to key out any specimens that I give you on the midterm exam, be able to identify them by the domain, kingdom, or phylum using the key that you have at the end of chapter 2. For that, in order to do that, you'll need to know all the terms associated with the key. So now I'm going to go over a couple of the terms and then practice keying out a specimen. First thing I want to mention is symmetry. There are 3 different types of symmetry we mentioned in lab. Symmetry is just the way that body parts are distributed about some sort of central point or central plane. The easiest kind of symmetry is no symmetry at all. Organisms are asymmetrical, like the sponge. It's asymmetrical because there's no place this sponge can be cut into 2 equal halves. So no symmetry at all, it's asymmetrical. Organisms that are bilaterally symmetrical, for example, like this crayfish, there's only one place the crayfish can be cut into 2 equal halves. That's right down the middle right here. You can't chop it here or here or here and get 2 roughly equal halves. So if there's only one way to cut it, it's bilaterally symmetrical. And human beings, as are most higher animals, are bilaterally symmetrical. Okay, the final type of symmetry is radial symmetry. Radially symmetrical organisms have more than one way they can be chopped into 2 equal halves. So starfish is a classic example. You could cut it here, you could cut it here, or you could cut it here into roughly equal halves. Also things like a jellyfish or a sea anemone are also radially symmetrical. Most organisms that are fairly round in shape are radially symmetrical. Okay, the next thing I wanted to go over is skeleton types. Classic kind of skeleton is calcareous endoskeleton. So we've got a deer skull here. So a calcareous endoskeleton is a hard skeleton inside the body. It's called calcareous because it's made of calcium compounds like calcium carbonate. However, there's some other kinds of skeletons other than hard ones inside the body. There are exoskeletons, which are skeletons outside the body. We've got 2 main types we're interested in. We have a calcareous exoskeleton, like it's composed of the seashell or mollusks. It's a hard skeleton made of calcium compounds outside the body. I'm sure all of you have probably stepped on a bug and you've heard it go crunch. So if you step on an animal and it goes crunch, it probably has a chitinous exoskeleton, like these cicada skeletons or exoskeletons. They are formed of a carbohydrate called chitin, and basically it's thin, it's flexible, and it's also crunchy. So I want you to remember chitinous exoskeletons and also calcareous exoskeletons. The third and final skeleton type is probably a skeleton type you've never even considered before. That's a hydrostatic skeleton. Hydrostatic skeletons are not hard at all. If the organism were to rot away, it would not really leave anything behind. Hydrostatic skeleton is composed only of body fluid under pressure, like this earthworm. You can think of a hydrostatic skeleton as being like a water balloon. Just that liquid under pressure in the body cavity gives the organism its shape, just like water inside a water balloon will give the water balloon its shape. Got a few more terms to mention. one is difference between, or rather, let's talk about multicellular organisms versus unicellular organisms. All living organisms are made of cells. A cell is just the smallest unit of life. Most cells are very small and microscopic, with a few exceptions, and they are surrounded by membrane. For example,